Welcome back to the AI Research Roundup. I'm your host, Alex. Today we're discussing multimodality representation learning for antibody-antigen interactions prediction from March 22, 2025. This paper aims to improve how we predict antibody-antigen interactions, or AAI. These antibody-antigen interactions are key to our immune response. A major hurdle is the scarcity of data combining molecular structure and sequence. So the researchers developed MuLAAIP, a new framework. It uses a multimodality approach, integrating both 3D structural and 1D sequence data. This allows it to learn detailed relationships using graph attention networks, AI that highlights important structural features, and normalized adaptive graph convolution networks to capture sequence similarities between antibodies. They've also created a new dataset for this work. Okay, and for those of you who like to dig into the code, the authors have made their work available on GitHub. In their MuLAAIP repository, you'll find the Python implementation, including scripts for processing the data, building the model itself, and, well, for training it, along with access to their benchmark dataset. Alright, this figure really helps visualize what we've been talking about. Part A shows their multimodality representation, essentially how they look at proteins from several different angles. You see, they capture details at the residue level, like the specific positions of atoms, then at the backbone and side chain level, focusing on their 3D orientations, and finally, at the whole protein level. Here, they use a protein language model, or PLM. These are sophisticated AI models trained on vast datasets of protein sequences, allowing them to learn the y-lengths of proteins and represent their sequences numerically. This numerical representation then helps form a graph of the protein. Then, Part B, that's the MuLAAIP framework itself. It shows how the sequence information and the structural data processed by those graph attention networks we mentioned earlier are handled in parallel for both the antibody and antigen. These different streams of information are then combined using multi-layer perceptrons, or MLPs. MLPs are a fundamental kind of neural network that, in this case, learn to weigh all these different features to predict important outcomes like binding affinity and neutralization. This framework really showcases their comprehensive approach to integrating diverse information. Alright, now this figure shows results from an ablation study, which is essentially where researchers test their model by removing specific components, one by one, to see how crucial each one is. We're looking at performance on different tasks like wild-type binding affinity and neutralization. For the affinity tasks, they use PCC, or Pearson Correlation Coefficient, a score from -1 to 1 indicating how well predictions match actual values, with 1 being a perfect match. For neutralization, it's MCC, or Matthews Correlation Coefficient, another score from -1 to 1 that's good for these yes-no type predictions. The key takeaway is that the full MuLAAIP model, the rightmost bar in each group, generally performs best. Removing any piece, like the structural information or the protein language model, usually lowers these scores, showing each part contributes significantly. Okay, so building on that idea that all parts of MuLAAIP are important, this table, Table 1, really drives that home by comparing MuLAAIP to other models on a specific task, predicting SARS-CoV-2 neutralization. They use several metrics here, including ACC or accuracy, which, just like it sounds, measures how often the model is correct, and the F1 score, a measure that considers both the precision of the predictions and how many true interactions are found. And as you can see, MuLAAIP, listed under co-modeling, consistently achieves top or near top scores across most of these metrics, including the F1 score and that Matthews correlation coefficient we talked about. This really highlights the strength of its combined approach. To wrap things up, it's clear that MuLAAIP's strength really comes from its ability to integrate diverse information, like sequence and structure from both antibodies and antigens. This kind of multimodal learning, where different types of data are combined, seems to be a powerful way forward for understanding these complex interactions, which could really speed up the development of new therapies. Well, that's all for today's AI Research Roundup. Thanks for tuning in, and I'm Alex.